Wow, this is a really long chapter, no girls? I've already sent the note because might be some of you want to go through, no? It's a very long note because it's a very long chapter, okay? So let's continue. Regarding the heterochromatin, it will be what? It will be the opposite of the euchromatin, no? It will be the dark stained region, okay? The euchromatin was the light stained region, remember? And the heterochromatin is your dark stained region. The chromatin remains in the condensed state and is called the heterochromatin, okay? So it's always condensed, okay? It's always squeezed together. In 1928, Heinz defined it as those regions of the chromosome that remain condensed during interphase and early prophase and form the so-called chromocenter. Okay, so they remain condensed. They don't open up and replicate, no? The heterochromatin is characterized by its high content of repetitive DNA sequences, okay, and contains very few structural genes. It is late replicating, that is, it is late replicating, that is, it is replicated when the bulk of the DNA has already been replicated, okay. So this is your backup to your backups, you know, like when you want to back up your phone, you know, you send it to the cloud. You want to back up to the cloud, you send it to the drive. Back up the drive, you put it in your pen drive. When you back up to the pen drive, you put your external hard drive. Like it's like a backup of a backup, no? Like security, like secure data, no? You will never lose it. It is not transcribed, okay? Because what is the point? No. Why should the DNA waste energy in undergoing transcription? Because it's not gonna create proteins from here. This is just the backup of the backup. Okay, so transcription is when you are, oh my god, so tired. When you are creating proteins, no, from the RNA. You will learn with Ms. D in detail. Okay, so let's continue. It is thought that in heterochromatin, the DNA is tightly packed in the 30 nanometer fiber. So the genes in the heterochromatin region are inactive. Okay. So during early and mid prophase stage, the heterochromatic regions are constituted into three structures, namely chromocenter, centromeres, and knobs. Okay. So during early and mid prophase, okay, the heterochromatin has three structures. Okay, it forms three structures: the chromomeres, centromere, and the knob. Chromomeres may not represent true heterochromatin since they are transcribed. Okay, so some of you may be thinking, no, if let's say this is a chromosome and that is a centromere, okay, like you must be thinking, no, when the spindle fibers pull apart, okay, into the opposite directions, when they pull apart, how come some of the genes are not damaged? So now you know, it's because the spindle fiber, they are very smart, no? They will not pull from the euchromatin part, you know? That is the DNA part which will replicate. No, it will not pull from there. It will pull from your heterochromatin part, you know? Heterochromatin is nothing but the junk DNA, you know? It will pull from there, okay? Because the heterochromatin forms the centromere. So simply, yeah, go ahead, pull, because it is not important. Okay. So you can see how nature has adapted itself. Okay, let's continue. Centromeric regions contain heterochromatin. So the centromere region contains heterochromatin. In salivary glands, these regions of all the chromosome fuse to form large heterochromatic mass known as the chromocenter. So those giant chromosomes, remember I showed you in the spotting. So those they become so giant because it's the fusion of so many centromeres. Okay, 1 minute, let me read again. In salivary glands, these regions of all chromosomes fuse to form large heterochromatic mass called as chromocenter. Okay, so it is the It is the unimportant part, okay, the heterochromatin part that fuse to form these giant mass, okay. Then knobs. Knobs are spherical heterochromatin bodies, usually several times the diameter of the concerned chromosome. So, knobs, they are, yes, they are heterochromatin, but they are spherical, okay. They are spherical in shape, and the diameter is large, okay, because if this is your normal chromosome and a knob is something like that, so obviously the diameter of this is bigger than that, you know, diameter of this knob is bigger than the diameter of the rest of the chromosome. Okay. We can see knobs in maize, okay, and we can observe them during the Pachytene stage in maize, okay. So these are your basic net or entrance question, no? Which stage you see the knobs? You will say, Miss, Pachytene stage, right? Okay. So looking at this diagram, this is your heterochromatin. I don't know if it's clear. Should I zoom in? Let me pause. Okay, looking at the heterochromatin, you will see they are the more stained. This is dark blue, this is light blue. Okay, they are more condensed. They are silent genes, okay. And gene poor. When we say gene poor, it means high AT content, okay. AT are like repetitive, you know, adenine and thymine, they are repetitive contents. And it stains darker because they are very much condensed, okay. Lots of DNA that is junk and it's just a copy of a copy of a copy. You just squeeze them together and you store it in somewhere. So the euchromatin, so it is less condensed. As you can see, this one is light blue in color and they express the genes, okay. And they are gene rich, means opposite of the one we saw, they will be rich in the guanine and cytosine content. And the stain is lighter, okay. Then this is your structure of the nucleus. Oh my. Okay, so you can see the nuclear pore, the nuclear envelope, the nucleolus, no? Then the nucleoplasm. It's like cytoplasm but inside the nucleus. Then the chromocenter. Chromocenter is what? Is your heterochromatin. Then the septum. Then you can see some euchromatin and some thick part, which is the heterochromatin. This one thick part, no, condensed part, and this is the thin part, this is the euchromatin. Then when we look at the structure of the heterochromatin and the euchromatin, you can see that the core histones are tightly packed. Okay, the histone proteins are tightly packed. We'll see about histones and all later. Linker histone we'll see later. Here it is less tightly packed, okay. So this is the stretch part of your DNA. This round beads, these are your histone proteins and those are the linker DNA. Okay. And it's simply histone only and the linker that is coiled. Depending on how tightly coiled they are, they will be heterochromatin. If they are more loosely coiled, they are euchromatin. So heterochromatins are of two types, the constitutive and the facultative.